So we're going to go over G protein-coupled receptors in a little more detail in this video. So, as we said before, the outside is the N-terminus and the inside is the C-terminus. And what we have right here is the amplifier. And normally, if we look at the subunits, it's usually alpha, beta, and gamma all bound together. And what happens in this form, since it's GDP bound, the moment it becomes GTP bound, as we see right here, the affinity for beta and gamma subunits become less and they start to drift down. So whenever it's GTP bound, we're now in the active phase. So this is the active, and this would be the inactive. And just remember that in the inactive form, it's still going to be bound to the GPCR right here. And so what happens is a ligand comes in, and it's going to bind to the receptor, and this is going to cause this change right here. And what allows GDP to go to GTP is something called a GEF. It's a GTP exchange factor. And if we want to go this way, we have a gap. So just remember that these two compounds are going to be doing the hydrolyzing and phosphorylating. So they're going to act as your kinase and your dephosphorator. And what we know so far is that we have different subunits that basically can come about. And so if we're looking at adrenaline, for instance, adrenaline, it goes under the adrenergic pathway. And we have the alpha-1 adrenaline, alpha-2, beta-1, and beta-2. And alpha-1 is related to GQ. And GQ is responsible for PLC beta increase. And what this does right here is we're going to have stimulation leading to the formation of IP3 and DAG. And if you remember, IP3 and DAG are involved with the release of calcium. So this is going to change the intracellular concentration of calcium, specifically IP3. So this would be increase in intracellular calcium concentration, and IP3 is basically going to interact with the sarcoplasmic reticulum to allow for this release of calcium. And DAG, it's kind of along the same path. So now let's look at alpha-2. Alpha-2 is involved with GI. And GI is going to cause adenyl cyclase to decrease, while GQ, I should have said, is going to increase adenyl cyclase. So GI, remember, it has an I, it deals with inhibition. So this also means CAMP is going to go down as well. Sorry, I made a mistake right here. This is not alpha adenyl cyclase. Sorry about that. Next we have beta-1 and beta-2, and both of these are involved with GS. GS is what increases adenyl cyclase. And as you probably guessed, its C-amp is going to increase as well. And you may ask, what the heck's the point of any of this? Well, these pathways are going to allow for either activation or I should say stimulation or inhibition in terms of your adrenaline and noradrenaline. Another pathway we also have is the acetylcholine pathway. And the acetylcholine pathway actually takes two routes. The acetylcholine, we have our nicotinic receptors, and then we have our muscarinic. Now, the nicotinic an example of this would be your NMDA receptors, which are found in the brain. They typically have 4 subunits, it's glutamate, and then you have the properties associated. So let's look at this a little in more detail. So if we have our acetylcholine right here, and we said we have 2 routes, the nicotinic and the muscarinic. So let's start with the nicotinic. The nicotinic is a 5 subunit cysteine loop, and it's ligand gated. And we've already learned that ligand gated is ionotropic, which means that it's going to be very fast. So some of the properties of the nicotinic are they bind acetylcholine. When they're open, you have sodium and calcium flowing into the cell, causing membrane depolarization, and that leads to an increased probability for an action potential. Now, if we're looking at the ligand gated, some of the properties are, as I said, it's fast. It's going to be in your excitable tissue. And they're going to regulate ion flow across the membrane. So we want to control the amount of ions going back and forth across the membrane, because if you have too much of one ion, you can result in hyperpolarization and other related problems. The nicotinic, as I said, this is going to be basically what's found in your skeletal muscles. And if you think about it, we would want them to be in the skeletal muscles because we're controlling ion concentration. We're also controlling the probability of an action potential. And we also know that acetylcholine is usually found at the receptors for the muscles. And so now let's look at the muscarinic. The muscarinic, it's found in inhibiting, or the function is inhibiting cardiac muscle. It's going to activate visceral smooth muscle. And this is an example of a GPCR. So G protein-coupled receptor. As we know with GPCR, what happens is the on-off is going to be dependent on the GTP or GDP bound state. So it's going to have an intrinsic GTPase to control GTP versus GDP. It's found in 80% of hormones. It's found that 80% of hormones are activated by G proteins. And as we've seen before, it is a 7-transmembrane receptor. So basically what happens is, if we were to look back at our image before, the first step that's going to happen is we're going to be GDP bound. And what will happen is Jeff will convert to the GTP state, giving us this state right here. And then what happens is the beta gamma subunits are going to dissociate from the alpha GTP bound. And I said this is because GTP has a higher affinity for the alpha subunit, causing a decreased affinity towards the beta gamma subunits. And this, once this happens, we're going to activate the amplifier right here. And now the signal is activated. And what happens if we look at the alpha subunit, it has the GTP GDP binding subunit. It's going to have GTPase activity. And if you think about it, if you're going to have something binding GTP GDP, you have to have a GTPase to control whether you're going to be in the active or inactive form. And then it's going to also have a lipid anchor. And the lipid anchor is going to be what holds it onto the membrane. The beta gamma subunit, it also has a lipid anchor, as you can tell. And the beta gamma itself, it's going to regulate downstream effectors. So once it goes to the amplifier, it regulates the downstream effectors. Some of the targets for beta gamma are going to be your ion channels, specifically potassium, adenyl cyclase, and phospholipase C beta. So that's some of the big key points about what's going on when it comes to this group. Now if we go back to our acetylcholine here, acetylcholine, much like what we saw with GPCR, it's going to have some effects. So it's going to be involved with GQ alpha, and it's also going to be involved with GI alpha. And we remember that the GI alpha form is going to be the inhibitory form. So it's going to inhibit adenyl cyclase. And so what that means is we're going to have inhibiting CN. So we expect that concentrations of adenyl cyclase are going to go down. Concentration of CAMP is going to go down as well. And basically this is what's involved, or the GI alpha is going to be involved in inhibiting your cardiac muscle. Now if we look at GQ alpha, what we said about this is that GQ alpha is related to phospholipase C beta. And this is specifically for the adrenergic receptor. Remember we saw this with alpha 1. And what this does, as we said, it activates PLC beta. It's going to increase the concentration. And so what's happening is PIP2 is basically making our DAG and our IP3. And we said before that IP3 is involved with calcium. concentrations. So this is going to be our signal transduction pathway. And then right here, our DAG is going to be a second messenger, which activates PKC. So I hope that makes sense to you guys. Now, if we want to terminate this whole process that we've seen, now we're going to have to use something else. So we can use two things to terminate the GPCR. One of the routes we can take is using GAP. Another route is using RAS. Or sorry, not RAS, RGS. And both of these terminate our GPCR. And so GAP, the actual name of it is GTPase activating protein. RGS is regulators of G protein signaling. So if you just remember what these two mean, you know that you're going to turn off your GPCR. Once off, then we go into this normal state where we're not worried about, you know, these other pathways of producing CAMP or decreasing CAMP or making calcium or causing the release of calcium, or, you know, any of these pathways that we've seen. Now, we have these two options right here with CAMP and CAMP regulation. So now the concern becomes, how do you turn off when you're making too much CAMP? So as a quick refresher, remember that G-alpha I and GS are both involved in CAMP production. So CAMP decreases by the inhibitor and CAMP increases by the stimulator. And so now, using different phosphodiesterases, we can control which what happens in the rest of the pathways. So termination of the CAMP and GMP pathways. So if we have CAMP and we've made too much, we can use phosphodiesterase 4, which is going to convert it to 5' AMP. And this basically means that we're going to decrease our amount, or we're going to control and regulate the amount of cyclic AMP produced. We also have phosphodiesterase 5, which does the breakdown of cGMP to make our 5' GMP. And we can also use phosphodiesterase 1. And phosphodiesterase is basically going to break down both CAMP and cGAMP. Now, if phosphodiesterase 4 is inhibited, this is going to cause an increase in cAMP, which leads to muscle relaxation and bronchioles. So this is ideal for patients with asthma. As I said, the PDE1 has a dual specificity. And if we inhibit phosphodiesterase 5, it's going to cause an increase in cGMP, which is basically what Viagra does. And so this would be the pharmaceutical grade Viagra. And the last thing that we need to go over is, what about our small monomeric G protein subunits. So we have GPCR and we have enzyme-like receptors. And these both are going to lead into small G proteins. And small G proteins are involved in proliferation, survival, cell movement, vesicular traffic, and secretion. The thing that is good to know, or the thing that's important about the small G proteins, is they're not directly linked to a receptor. And same as before, the GTP is the active state, GDP is the inactive state. The small G proteins are monomeric, and they're going to be involved with RAS and Rho. And RAS and Rho, or RAS specifically, was originally identified as the oncogenes in human tumors. And same as what we saw before with Jeff and GAP, they are going to control the GTP versus the GDP states. So everything we saw before is also applicable in this. Well, if you have any questions, leave it in the comments below. If you'd like to download my notes, I've left it in the caption. Please subscribe to the channel.